Right now, as you read this, microscopic 8-legged arthropods are living inside your face. They mate at the openings of your pores, lay eggs in your follicles, and complete their entire life cycle in about 2 weeks without you ever feeling a thing. This isn't science fiction, and it's not a hygiene problem. It's just being human. Today, we're breaking down the most unsettling discoveries about the human body, from the mites crawling on your eyelashes to conditions that literally lock you awake until you die, starting with the tiny roommates you never agreed to host. Demodex folliculorum. Demodex follicularum is a mite species that lives exclusively on human skin, so adapted to you that it dies within hours if removed. It sets up shop in your hair follicles and sebaceous glands, feeding on sebum and dead skin cells while you go about your day completely unaware. The life cycle is 14 days. Egg to adult. Mites mate at the follicular opening, retreat down into the pore to lay eggs, and the whole cycle repeats without ever leaving your face. Their entire world is your face. Before you start scrubbing, here's the thing. Everyone has them. You probably picked up your first mites as an infant through skin contact with your mother, and they've been there ever since. Fewer than 5 mites per square centimeter is considered normal. You're walking around right now with a low-level arthropod civilization in your pores, and your body is completely fine with it until it isn't. When populations explode past 60 per square centimeter, problems start. Inflamed eyelids, rough scaly skin, and on the scalp, chronic infestation has been linked to hair loss caused by enzyme-driven inflammation, slowly miniaturizing the follicle until growth stops. But for most people, nothing happens. The mites just live there, quiet, invisible. You are never truly alone, not even on your own skin. At least Demodex stays on the surface. The next discovery is written directly into your DNA: human endogenous retroviruses. So, face mites are weird, but at least they stay on your skin. Here's something deeper. Roughly 8% of your entire genome is composed of ancient viral DNA that integrated into your ancestors millions of years ago and has been passed down ever since. You are, at a fundamental level, partly built from fossilized infections. Human endogenous retroviruses, or EARVs, are genomic relics of retroviruses that invaded the germline of distant primates, got stuck in their chromosomes, and became hereditary. Structurally intact RV sequences mirror modern retroviruses like HIV. They carry recognizable GAG, POL, and ENV genes flanked by long terminal repeats. Most have been disabled by mutations over evolutionary time, but members of the HERVK superfamily can still produce retrovirus-like particles in certain contexts. These aren't theoretical fossils. They're DNA sequences in every single one of your cells that retain the molecular signature of functional viruses. But here's where it gets complicated. Some HERV sequences have been co-opted as functional biology. Certain HERV envelope genes became the basis for proteins that mediate cell fusion and placental development. Viral invaders of the distant past are now embedded in how humans reproduce. The boundary between self and invader dissolves at the chromosomal level, and it's not all ancient history. Aberrant expression of specific HERV families has been observed in germ cell tumors, some leukemias, and autoimmune conditions including multiple sclerosis and systemic lupus erythematosus. Proposed mechanisms include molecular mimicry, where viral epitopes share amino acid motifs with host proteins, leading to cross-reactive immune responses. Also, superantigen activity, where HERV products bypass normal antigen processing and activate large subsets of T cells indiscriminately. Your genome is not a pristine human manuscript. It is a palimpsest with chapters written by viruses that colonized ancestors across primate evolution. But if you think carrying viral DNA is strange, wait until you hear about the cells from other people living inside your body for decades. Microchimerism. Your body is not one person at the cellular level. It's a mosaic, and a portion of those cells might be genetically someone else's. During pregnancy, cells cross the placental barrier in both directions. Maternal cells migrate into the fetus, fetal cells migrate into the mother, and both populations can persist for decades. This is microchimerism, and it's not rare. Many women carry a measurable population of cells from their children for the rest of their lives. These cells aren't dormant tourists. They populate the heart, liver, and brain, integrating into tissue, and potentially influencing immune responses indefinitely. Here's where it gets genuinely strange. Researchers have found T cells in fetal lymph nodes that specifically recognize maternal antigens and actively suppress immune responses against them. Your immune system doesn't just encounter your mother's cells, it gets programmed to tolerate her, which explains why, if you received a kidney from your mother, your body might accept it better than a stranger's. You've been carrying her cells since before you were born, but the outcome depends entirely on timing. Fetal exposure to maternal cells during immune development produces lifelong tolerance. A mother's exposure to fetal cells in adulthood more often produces sensitization. Same cells, opposite outcomes, purely based on developmental timing. Fetal microchimerism has also been implicated in autoimmune conditions like systemic sclerosis, though whether these cells are causing damage or attempting repair is still debated. What unsettles people most is the poetic reality. My mother is always with me is not metaphor, it's measurable, cellular fact. The boundary between self and other dissolves at the cellular level, replaced by a mosaic identity that persists long after pregnancy ends. But if you think carrying your mother's cells is unsettling, wait until you hear about the microbes in your gut that might be shaping your mood, appetite, and even memory. The gut-brain axis. Your mood today might be partly a product of what bacteria are thriving in your gut right now. Before you roll your eyes, hear me out. The gut-brain axis is a bidirectional communication network linking the gastrointestinal tract and its resident microbes to the central nervous system via the vagus nerve, the hypothalamic, pituitary, adrenal axis, immune, cytokine networks, and metabolic pathways. And that's not hyperbole. That's neuroanatomy. The vagus nerve is essentially a biological highway running between your gut and your brain. When you eat, gut microbes produce short-chain fatty acids like acetate, propionate, and butyrate that can modulate neuroplasticity and gene expression in the brain. They also participate in the synthesis of GABA, serotonin, and dopamine, and generate neuroactive metabolites including D-lactic acid and ammonia. Glucose entering the gut triggers GLP-1 release, which activates the nodose ganglion, and via vagal pathways engages brain regions associated with memory encoding, the hippocampus and amygdala. The act of eating sends precise neural signals that can influence how we form and recall memories. In germ-free mice, exaggerated stress responses are observed with elevated plasma ACTH and corticosterone compared to normal controls. Colonizing these animals with specific microbiota can normalize these HPA axis responses. And vagotomy abolishes some behavioral effects of probiotic strains, confirming the vagus nerve as a critical conduit. The vagus nerve carries not just instructions from brain to gut, but ongoing sensory information from the gut to central circuits involved in interoception and affect. The brain's sense of how the body feels is being continuously updated by microbial and nutritional status in real time, which raises an uncomfortable question. If feelings you experience as authentically yours are being co-authored by microbes that have no stake in your well-being, just their own, then where exactly does you begin and them end? At least the microbes stay in your intestines. Unlike what we're about to cover next, teratomas. There are tumors that grow teeth inside your body, hair, eyeballs, bone fragments, fully formed tissue types that have no business being where they are. And I can prove it. Teratomas are germ cell tumors that arise when pluripotent cells, the kind that can become anything during development, escape normal developmental cues and start differentiating in completely the wrong place. They produce tissues from all three embryonic germ layers, ectoderm, mesoderm, and endoderm, in disorganized chaotic arrangements inside adult bodies. You can open an ovarian cyst and find mature skin, hair mats, teeth embedded in the wall, cartilage, bone nodules, and glandular structures. It's embryogenesis running without an instruction manual. But here's where it gets genuinely disturbing. Experimental data shows how context-dependent this process is. When human embryonic stem cells were implanted subcutaneously in immunodeficient mice, only 25 to 40% formed teratomas. But when co-injected with Matrigel, a basement membrane matrix, efficiency shot up to 80 to 100%. Under the kidney capsule, 100% of implants formed teratomas. The extracellular environment determines whether developmental potential becomes ordered regeneration or aggressive tumor growth containing fragments of a body. And it gets worse. Ovarian teratomas containing neural tissue can express NMDA receptors, the same receptors in your brain. Your immune system sees these receptors in the tumor, learns to attack them, and then cross-reacts with the identical receptors in your actual brain, causing anti-NMDA receptor encephalitis. A tumor growing in your pelvis produces psychiatric collapse and seizures originating in your skull. Your body teaches your immune system to destroy your own mind. The same pluripotent properties that make embryonic stem cells promising for regenerative medicine make them inherently tumorigenic, if not precisely controlled. The line between therapeutic tissue regeneration and a cyst containing teeth is purely a matter of microenvironmental context. At least teratomas stay in one place, the next condition doesn't. It spreads through your muscles, slowly turning them into bone until you can't move at all. Fibrodysplasia, ossificans progressiva. Your body contains a blueprint for building a skeleton. In fibrodysplasia ossificans progressiva, that blueprint gets the wrong address. A single point mutation in the ACVR1 gene turns muscle into bone, piece by piece, injury by injury, until you're locked inside a second skeleton you never asked for. Here's the thing, this isn't calcification. This isn't your muscles hardening or filling with calcium deposits. This is real bone, built correctly, using the exact same program that constructed your original skeleton in the womb. The mutation hijacks a BMP receptor, making it hyperactive. Every time you get hurt, your body initiates repair. But the repair machinery has been reprogrammed. The cellular culprits are muscle-resident mesenchymal cells, called fibroadipogenic progenitors. When they carry the ACVR1 mutation, they stop supporting muscle repair and become ossification factories instead. They form cartilage templates first, then replace them with lamellar bone, following the full endochondral sequence. Textbook development, just happening in your bicep instead of your femur. But it gets worse. Mutant cells don't just differentiate wrong. They actively suppress normal muscle repair by shutting down the cells that should be rebuilding your muscle. Each injury not only heals incorrectly, it makes future ossification more likely. The mutation doesn't just add bone, it dismantles the alternative. AYAP typically announces itself early. Many patients are born with malformed great toes, a telltale sign that skeletal development is already off. Then the flare-ups begin. A bump, a fall, a viral illness, an intramuscular injection. What should be minor becomes catastrophic. Painful soft tissue swelling appears, then ossifies over weeks. Spine, shoulders, hips, jaw, cumulatively fused. Respiratory failure from chest wall rigidity is a leading cause of death. Global prevalence is roughly 1 in 1 to 2 million. And here's the cruelest detail. Trying to surgically remove the ectopic bone just triggers more ossification at the surgical site. The body reads surgery as trauma and responds with more bone. There is no way to fight back without making it worse. At least FOP patients retain sensation and motor intent, unlike what's coming next. Because phantom limb syndrome will show you that the brain doesn't need a body part to insist it's still there and still hurting. Phantom limb syndrome. Your brain is so committed to the idea of your arm that even after it's gone, the brain insists it's there, and it can make it hurt. Phantom limb syndrome is the persistent perception of a limb that has been amputated, including vivid sensations of position, movement, temperature, and pain arising from a mismatch between motor intentions and absent sensory feedback, combined with cortical reorganization in somatosensory areas. Functional imaging studies show that attempts to move a phantom limb activate sensoromotor circuits, including the parietal cortex, in patterns closely resembling those of intact limb movement. Painful phantom sensations are associated with activation of parietal regions that integrate motor commands with expected sensory feedback, regions that keep firing even when the expected feedback never arrives. Here's where it gets weird. Cortical reorganization following amputation allows neighboring somatosensory representations to invade the cortical territory of the missing limb, producing phenomena like referred sensations. Touch a patient's face and it can trigger vivid sensation in a phantom hand. The brain's body map is plastic and it's unreliable. Phantom limb pain affects an estimated 60 to 80% of amputees and can be severely disabling. Some patients experience a phantom hand locked in a clenched fist they cannot open, a position of permanent, inescapable pain generated entirely by neural prediction in the absence of any physical tissue. The limb is gone, but the brain refuses to update the map, and the result is agony with no source. Mirror therapy, in which visual feedback from the intact limb is reflected to appear as the missing one, can reduce phantom pain in some patients by resolving the motor sensory mismatch. The brain's pain response can be partially corrected by feeding it a convincing visual lie. You can trick the brain into letting go of a limb it insists is still there by showing it a reflection in a mirror, which tells you everything you need to know about how much of your sense of your own body is a controlled hallucination. But if you think a phantom limb is disturbing, wait until you hear about a disease where a single misfolded protein erases your brain from the inside out with no cure, no treatment, and no escape. Prion disease. Your brain can be destroyed by a protein that isn't alive, doesn't have DNA, and reproduces itself by forcing other proteins to copy its shape. And I can prove it. Prion diseases are fatal neurodegenerative disorders caused by a mechanism that shouldn't be possible. A misfolded host protein called PRP-Res propagates by physically templating its abnormal conformation onto the normal cellular form. PRP-SEN, spreading without any nucleic acid. The infectivity, the strain identity, the entire pathogenic program is encoded not in genes, but in three-dimensional protein architecture. The critical conversion region spans roughly residues 108 to 189 of the prion protein, encompassing the core folded domain. When PRP misfolds, it acquires substantially higher beta sheet content, and the specific conformation of that beta sheet varies across different prion strains. This means that strain phenotypes, including incubation period, lesion distribution, and clinical presentation, are encoded not in DNA, but in protein shape. Here's where it gets worse. Prion strains can be operationally distinguished by distinct incubation periods and neuropathological patterns, even when generated from identical PRP amino acid sequences. Conformation alone constitutes a primitive hereditary system capable of templated replication and strain-level evolution under selective pressure. Information exists in a fold. The protein remembers how to kill. Clinically, human prion diseases include sporadic Creutzfeldt-Jakob disease, familial forms caused by PRNP mutations, variant CJD linked to bovine spongiform encephalopathy exposure, and iatrogenic transmission via contaminated medical instruments. Patients experience rapidly progressive dementia, ataxia, myoclonus, and psychiatric symptoms. The brain develops widespread spongiform vacuolation, microscopic holes riddling the neuropil, punching through neural tissue until the architecture of thought collapses entirely. Progression is typically rapid, months to a few years from symptom onset to death. There is no cure, and prions are notably resistant to conventional sterilization methods, including standard autoclaving. They can persist in the environment and on surgical instruments, requiring specific combinations of high temperature, strong chemical agents, and extended treatment times for reliable inactivation. A misfolded protein can survive conditions that destroy bacteria, viruses, and most other biological threats. But if you think the general concept of prion disease is disturbing, wait until you hear about the one specific mutation that selectively destroys your ability to sleep. Fatal familial insomnia. Fatal familial insomnia is exactly what it sounds like. And being exactly what it sounds like does not make it less devastating when examined in clinical detail. It is a prion disease caused by a mutation in the PRNP gene that produces progressive and ultimately complete destruction of the thalamus, the brain structure responsible for, among other things, regulating sleep. The disease presents first as insomnia, then as a total inability to sleep, then as dementia, hallucinations, and autonomic dysfunction, then as death. The entire progression from first symptom to death typically takes between 7 and 73 months. It is inherited in an autosomal dominant pattern, which means having one copy of the mutation is sufficient. It means that if your parent has it, you have a 50% chance of having it too. The biological reality is that the thalamic destruction produced by FFI's prion accumulation is progressive and selective. Other brain regions are affected, but the thalamus is the primary target, and its deterioration produces the specific pattern of sleep dysregulation that defines the disease. The thalamus gates sensory information to the cortex and plays a central role in generating sleep cycles. As it is destroyed, sleep cycles become fragmented, then non-existent. The brain loses the ability to perform the neurological function that sleep exists to provide, while the rest of the patient's cognition remains intact enough in the early stages to be fully aware of what is happening. The existential horror is the awareness. Early-stage FFI patients know their diagnosis. They experience the insomnia as conscious, continuous wakefulness they cannot escape. Not unconsciousness, not ignorance, but awareness of every hour they cannot sleep. Sleep deprivation at this scale is itself torture. Hallucinations appear. Cognition fragments. The patient remains aware of their own deterioration for as long as awareness is possible, and there is nothing that can be done about it. Sedatives cannot produce sleep once thalamic destruction is sufficiently advanced. The brain has lost the structure sleep requires. The clinical manifestation progresses through 4 stages: progressive insomnia with panic attacks, escalating hallucinations and anxiety, complete inability to sleep with rapid cognitive decline, and finally total dementia, followed by death. There is no approved treatment. Families carrying the mutation can undergo genetic testing. Having that test returned positive means knowing with certainty what is coming, and knowing in equal certainty that knowing changes nothing about how it arrives. If you enjoyed this video and want to see more, click the video on the screen now.